So we have the first talk given by Rodrigo Siqueira de Souza from University of Sheffield, and he is going to talk about FootSim, modeling tactile responses from the sole of the foot. It's all over to you.
So yeah, hello there. As I was kindly introduced by Adele, my name is Rodrigo. I work at the University of Sheffield as a research associate. And today we're going to be talking about the tactile responses of the foot sole. But why are those tactile responses important? Well, they're important mostly for gait and for balance. So when we have a normal subject on an experiment walking and we use anesthetics on their foot sole, like we can see on those plots over here, the velocity of gait, which is the y-axis of the plot, gets significantly reduced, as you can see at the gray bar in opposition to the black bar, when you anesthetize the foot. The opposite is also true. If you have patients with suffered stroke or they have diabetes, if you put vibrating insoles on their shoes, you will really reduce the postural sway that you have on those conditions. So the tactile feedback from the foot sole is very important for walking and gait. But where does it come from? Where does information come from? Well, most of this information that we have for tactile afferents came from the hand, but we have 4 classes of tactile afferents. They are basically divided in how fast or slow they adapt to stimuli. So you have slow adapting and rapid adapting, and you have types 1 and types 2, depending on the sizes of the receptive fields. So small receptive fields type 1, large receptive fields type 2. So you can see on this color image here, the layers of the skin, the epidermis and the dermis, and where those receptors are placed. You have the Pacinian corpuscles, which are the RA2s over here, and the Messina corpuscles, which are the RA1s, which are thought to be the ones that have most importance for tactile feedback on the foot sole. But how do you study those afferents? Well, one way is to use microneurography, like our collaborators Leah and Nick did on Guelph University. They got participants to sit on and get electrodes implanted on their nerve, and then they recorded one afferent at a time. It's actually a very hard to do technique. It takes time. The subject has to be immobilized, and then they applied a range of amplitudes and frequencies of stimulation to the sole of the foot, a range that you would have in normal conditions like walk, from 0.0 millimeters to 2 millimeters, and frequencies ranging from 3 hertz to 250 hertz. And then they recorded this data set. So using that data, we built a model that takes into consideration the specific skin mechanics of the foot and deliver pressures to a quasi-static model of the skin that then is given to integrating fire neurons that have 12 parameters that you can interchange, a low pass filter. All of those 2 to 7 are time varying parameters. And then you integrate all of those into a saturation parameter because of the adaptation of those tactile afferents. Then you have a couple more post-inhibition parameters to account for refractoriness of the receptors. Also, as I mentioned, the skin of the foot is not like the skin of the hand. It's not homogeneous. So you have, as you can see on this blue and green foot sole plots, you have harder regions, which are the darker colors, and softer regions, which are the lighter colors. The same goes for thicker regions and thinner regions. So the heel is always the harder and thicker region versus the archer, which is softer and thinner. Also, the density of each one of those afferents varies in the skin. And this data also came from the microneurography study. So you have the density for each one of those afferent classes. So then we put the model to the test. Is our model firing the way those afferents were firing in the microneurography study, in the empirical one? So in this plot, you have on the y-axis the way our model fired for the combinations of amplitudes and frequencies that they did also on the empirical experiment. And on the x-axis, you have the firing of the experiment per se. So you can see that for the FAs, the top ones, the blue and the orange ones, as you can see in the legend, we are pretty much in the ballpark of what they should be. The assays are not so good. The reason for that being that for SA2s, we only had very few models to use to optimize the parameters on the fitting procedure that we did using differential evolution. And also our model doesn't account for stretch as well, and that's the main thing that SA2s are known for, one-off. So putting that to the test, we also went to the thresholds, different firing thresholds, which are the minimum amplitude per frequency that lists a certain firing rate, a certain number of spikes. So here you have on the y-axis the amplitude, and on the x-axis the frequencies of each one of the models, which are the individual lines, and the thresholds. Top 4 panels on the model, bottom 4 panels, the empirical ones. So they look quite similar. So we were pretty happy, and oh yeah, the model is behaving like it should be. So let's clump those models, group them per class. So if we group those per class, you can see those two graphs on the left. On the left, they are panel A is what came from the microneurography experiment. So we can see those thresholds there for each one of the classes, same color code as before. Panel B is our own model. And then we looked into them, they look very similar. So we went and measured the RMSAs, which is a measure of difference in between those two curves. And if you look at this graph on the right, you can see that the FAs, 0 there mean the curves are really similar. We are in the optimal scenario, and 1 being the worst possible case, that the FAs behave much better than the SAs, and they are all in a good range. The scatter points are actually the individual models over there. So it seems that everything is going well, that the model is behaving the same way as the afferents on the actual foot sole of both subjects behaved. So we decided to go even further and put the model to the test. We had subjects on the Insigno Institute done by our collaborator Claudia, putting sensorized insoles on their shoes, which are basically pressure sensitive insoles that can get pressure off your feet when you're walking or when you're doing any activity. And then using those pressures, we converted those pressures into indentations. That's what our model used, because as you remember, microneurography can only be done with patients immobilized and one afferent at a time. So now we can take these pressures and turn them into indentations, feed them back into the model, and do experiments that would not be possible in that scenario. And then we found out of the bat that for all the phases of the step, like the initial contact, the flattening of the foot, the mid stance, and the push off, you have different activations of each one of those afferent classes. So if you look at the bottom of the image, you have 2 foot soles paired. The red ones are the heat maps of the pressure sensitive insoles. So you can see how the pressure is distributed in each one of those parts of the step. The other foot, the one in black, is the activation of the actual neurons, the actual afferents. And we can see that on the initial contact, there is a burst of FA2 firing, which was something that was not known. Granted that it was a proof of concept experiment, was not that well controlled, but is a very good indication that we can actually use the model to experiment in ways that wouldn't be possible using microneurography. And then just to show you a small animation of that step happening, same as before, the heat map of the pressures on the left-hand side and the activation of the mechanoreceptive, the tactile afferents on the right-hand side. And you can see here again, the plot of those firing rates per time, and you can see the bursts in FA2 firing in the initial phases of it. And then this model is already being applied to prosthetics development by our collaborators on ATH Zurich, Natalija and Stanisa. So they were cover of Nature a while back. They are doing real feedback prosthetics by having sensorized insoles, giving stimulus to the nerve by using implants. And then using the things that we are optimizing in the now model, we aim on giving actually a good account of what the sensation would be for each one of those tactile afferents in the foot sole and restore the sensation on those subjects. So that's basically it. This is the team, Hannes, myself, and Luke and Claudia worked on the insoles, the guys on the ATH, Natalija and Stania, and Nicholas and Leah that did the microneurography experiments. So thank you very much for watching, and you can fire any questions you want.
Okay, so that's great. So thank you. That was perfectly on time. And yeah, so please ask any questions by typing them into Q&A box. So anyone who has questions, panelists or the audience, please type this into Q&A box. While people are thinking about it, maybe I could start. So I probably missed it. So what was these? So you had 4 different FA1, FA2, SA1, and SA2. So these are 4. So what they were, how they were different from each other? So what they're measuring?
So we got the data from the microneurography experiments, and then our model has the integrating fire neurons with 12 parameters. So we applied differential evolution to those models to try to fit them to the firing, or if each one of the afferents that were recorded in the experimental setting. So basically the same number of afferents that they recorded, we have it as models. So the total was about 52 models divided per class, a certain number of them in each of the classes. And then that's why I just showed you a representative image. Otherwise, I would show you a massive scatter plot full of information.
Yeah,
and
of course.
Then I clumped them into classes. That's why you only saw 4 classes, but those 4 classes actually came.
And how do you do that? Did you run some sort of a dimension reduction, or?
Sorry, I didn't catch that.
So how did you do that? So you said you had quite a lot of different, bunch of things, and from this you pull this out. So how do you do that?
So we had the firing rates for those experiments. So we knew that amplitude 2 and frequency 3 hertz elicited 50 hertz firing rate. So we had the responses of those afferents, and then we threw them into a machine learning differential evolution algorithm to basically tell me which combination of the parameters on that differential equation, which is the leak and fire model, would provide me with a response that was closer to the actual empirical one. That's how we did it.
Okay, that makes sense. Okay, so that's great. So I think we are going on time. So thanks a lot, Rodrigo, again. It was great talk. You're welcome. And so next we should crack on.